Hi, I'm Dr. Agnes and I'll be talking about my favourite topic today, and that's the gut microbiota. And I'll be looking at how it communicates with the skin and how we can harness this connection as a preventative measure in the cosmetic setting. I'll start with my disclosure. I'm a cosmetic physician and managing director of bioaesthetics, and I have a financial interest in Biome MD, which is a program I developed that's available to cosmetic clinics. What I'd like to go through today is firstly talk about how some of the common conditions that we see in our practice are related to the health of our gut. And I'll also then look at mechanisms through which the gut microbiota communicates with the skin under physiological and pathological conditions. And I'll finish up by looking at some evidence-based recommendations that we can make that have been shown to improve gut health and may potentially improve outcomes for our patients. So as you know, chronic conditions are on the rise, and that means that our patients are becoming increasingly complex, and this increases their risk of an adverse event. So the question is, can we address these conditions systemically in a way that's reasonable and appropriate within our scope of practice to help improve outcomes? Our top priority is to ensure patient safety, and this table is from an article that I highly recommend to all injectors. It outlines the steps we can take to avoid dermal filler-related complications, and it begins with identifying those at higher risk. And as you can see, the focus is on skin and systemic conditions or events that are associated with increased or altered immune activity. These are just some of the complications that we're wanting to prevent, and I won't go into detail, but I just wanted to point out that all but the vascular events are immune-mediated and more likely to occur in patients with disordered immune function. And this table shows the prevalence of these conditions that we routinely screen for. I've just added a couple of extra metabolic diseases because they're associated with systemic inflammation and they increase the risk of infections. So some of these conditions are quite common, and it's highly likely that we regularly see these higher risk patients in our practice. Now, over the last decade or so, what the research into the gut microbiome is finding is that dysbiosis, which is an altered state of microbial composition, and it is driven by dietary and environmental factors, is inducing intestinal and systemic inflammation and disrupting a number of physiological processes that ultimately increase the risk of developing these various diseases. One thing I did want to point out is that chronic inflammation is also linked to accelerated aging through this cascade of cumulative cellular damage that eventually leads to the external signs of aging, such as wrinkles, which is what most patients will present with. But you can see that there may be many other issues just beneath the surface. So to better understand how this connection works, I'm going to start with a crash course in the gut microbiome. So firstly, some terminology. Microbiota refers to microbial cells, whereas microbiome refers to their genetic material, and there's about 150 times more genetic material than that of the human genome. So their metabolic capacity far outweighs ours. And what the research shows is that the microbiome encodes for over 200 metabolites that have the ability to influence almost every single aspect of our physiology. There are different microbial communities in different parts of the bowel, and what I'll be referring to today is those that are found in the large bowel. And there are 5 main phyla there, and they contain a variety of species within them, and they have functional similarities as well. And what's important for health is that these phyla are maintained within specific proportions, and that there is a large diversity within each of the phyla, because that then translates to a better metabolic capacity. The key role of the microbiome is to digest dietary fibres that have escaped digestion by human enzymes. The microbiome has up to 100,000 genes for that purpose, whereas the human genome only has 17 genes. So dietary fibre is crucially important for their survival. It really is their main food source, and a lot of really beneficial species rely on it. What they do with this fibre is predominantly they convert it into short-chain fatty acids, which then act through specific receptors on our cells or through epigenetic regulation to influence the function of our cells. And these benefits extend well beyond the gut. So all of these microbial metabolites that you see here in the centre, such as enzymes, peptides, vitamins, and neurotransmitters, they have the ability to influence metabolic, immune, and neuroendocrine function within all of these organs. And there's another group of really important microbial products which are called microbe-associated molecular patterns, and these are cellular components of commensal species and of pathobionts, and they're released when the cell dies. Pathobionts are gram-negative bacteria that have beneficial functions when they're kept in low numbers, but they become pathogenic when their numbers increase, and that's what happens in dysbiosis. There are two examples of molecular patterns that have been extensively researched. That's lipopolysaccharide and flagellins, and they're associated with a number of inflammatory conditions because they have the ability to activate specific receptors on our cells that initiate intracellular proteins and then produce a potently inflammatory response, not just within the gut but right throughout the body, and they've been found within various organs as well. So when it comes to what happens in the gut, the main role of the microbiome is to protect us from the external environment because the gastrointestinal tract is a barrier from the outside, just like the skin is. A diet that's high in fibre and polyphenols will produce a really healthy and diverse microbial community, and a large amount of short-chain fatty acids that then go on to perform a range of functions that prevent the translocation of pathogens and antigens through the gut wall. The way they do that is, firstly, they help to maintain the integrity of the gut wall by providing energy to enterocytes and stimulating the synthesis of tight junctions and the mucus layer, and also the production of proteins and IgA that help to neutralize pathogens. Fatty acids also influence the innate and the adaptive immune systems, and it's estimated that about 80% of our immune cells are actually located along the length of the gastrointestinal tract within Peyer's patches and mesenteric lymph nodes, and also between enterocytes. And short-chain fatty acids are able to communicate with these immune cells directly. What they do is they can inhibit macrophage activation and direct dendritic cells to produce anti-inflammatory cytokines. And these cytokines then guide T cell differentiation towards regulatory T cells. And this is really important because these cells are anti-inflammatory. So all of these functions combined help to maintain a state of immune tolerance within the gut, where antigens that cross through the gut wall are rapidly managed without causing damage. And these products then become immune signaling molecules that can enter the bloodstream and convey immune tolerance in distal organs. So these effects have been found in adipose tissue, in the liver, in the brain, and also in the skin. And that's really what forms the basis between the communication between the gut and the skin under physiological conditions. So when there's a high microbial diversity, the immune, neuroendocrine, and metabolic signals produced will influence beneficial changes in the skin. And as you can see, those changes, they'll also influence immune tolerance within the skin and improve microbial diversity, and this all helps to contribute to homeostasis within the skin. Another thing that they do, these immune signals that are derived from the gut microbiota do, is they can influence T cell differentiation that enables the skin to adapt to disruptive stimuli. And this is really relevant in the cosmetic practice because dermal fillers are considered adjuvants that disrupt homeostasis. So potentially by optimizing T cell differentiation in the gut, we may be able to reduce reactivity and improve allostasis in the skin. This table shows some of the microbial species that produce these signaling molecules and have a direct effect on the skin. Another really important factor is this tridirectional communication between the gut, skin, and brain, and this appears to play a role in a number of common skin conditions like acne and psoriasis because of the activation of the HPA axis, which then disrupts microbial composition. So moving on to what happens in pathological states, there's a huge amount of research linking gut diseases to skin lesions, and I'll just quickly go through some of the examples, and you can see here that 25% of those with celiac disease will have some form of skin lesions. The same goes with those for those with inflammatory bowel disease, with psoriasis being the most common. And dysbiosis in the small intestine is associated with rosacea. And in this study, the rosacea resolved with antibiotic treatment of the dysbiosis. So I'll now look at how these pathological processes in the gut are associated with a range of skin manifestations and also some common chronic conditions that we see in our practice and are considered contraindications or precautions. So in states of dysbiosis, there is a reduction in protective short-chain fatty acids and an increase in molecular patterns from pathobionts and danger signals from damaged tissue. And these increase the permeability of the gut wall, which means that antigens and pathogens and these signaling molecules can then pass through. And these signals will then produce cytokines that shift T cell differentiation towards these effector T cells, which are Th1, 2, and 17, which are highly pro-inflammatory, and they cause inflammation within the gut. There are also changes in microbial neuroendocrine and metabolic output, and for example, with an increase in potentially damaging neurotransmitters from E. coli, and also there are changes in macronutrient and micronutrient processing within the gut, and that's associated with poor glycemic control and nutrient deficiencies, and these present with a range of skin symptoms. So these signaling molecules ultimately disrupt skin function and cause inflammation within the skin. And this image shows the changes that occur in acne when inflammation and oxidative stress and poor glycemic control increase the lipid content, and the increase in free fatty acids is detected as danger signals, which actually perpetuates the inflammation. And there's also an increase in colonization with P. acne. This image shows the link between dysbiosis and atopic dermatitis, which is characterized by a Th2 response. So the direction of T cell differentiation in the gut and in the skin and in distal organs will determine what conditions develop. And this differentiation depends on the individual's microbial composition and the signaling molecules that are produced, and also to some degree on genetic predisposition. So under physiological conditions, short-chain fatty acids will drive the regulatory T cell response to produce immune tolerance, and that's really what we want. Under pathological conditions, though, dysbiosis will commonly produce a Th1 response that causes inflammation in the gut, and that can become systemic, and if left unchecked, then these chronic inflammatory conditions can develop. One thing I did want to point out is that there's a really clear link between gut disorders and mental illness. And those with mental illness, one study showed that they appear to be more likely to seek cosmetic procedures and more likely to be dissatisfied with the results. So it might be that by correcting dysbiosis and reducing inflammation in the brain, we may be able to help improve patient satisfaction as well. So in those with genetic predisposition, there is a shift towards a Th17 response, which is associated with autoimmune conditions. And those with atopic disease see a shift towards a Th2 response, which we saw in the previous slide with atopic dermatitis. So these conditions, these differentiation pathways can coexist, and they often do, and all of them can contribute to accelerated aging. This table just shows the microbial species and T cell responses with three conditions, with rheumatoid arthritis, type 1 diabetes, and asthma. And you can see there are lots of references there, but there really is absolutely no shortage of published research that presents similar findings for almost every autoimmune and allergic condition. The relevance in the cosmetic setting is that these T cell related conditions place the patient at a higher risk of adverse events, and some of them are contraindications to the cosmetic treatments that we perform. One thing I did want to note is that the Th2 response is associated with an increase in IgE and histamine, and also with hypersensitivity reactions. But this increase in IgE and histamine may actually make the patient more sensitive to condensed protein powders like collagen, and also to fermented foods. Both collagen and fermented foods are very high in histamine. So I know that goes against what everyone does. It's commonly these foods are recommended for gut health, but they can make things worse for up to 40% of the population, and they're best avoided in the cosmetic setting. So the question is, would the use of gut protocols improve outcomes in cosmetic patients? And I don't have the answer to that question because there isn't any research in that respect. But there is a lot of research that links certain dietary and lifestyle modifications and also certain supplements to improvements in microbial composition and immune function. And these are really safe measures that might actually help reduce reactivity in the skin. So what I'll finish up with now is just some simple evidence-based recommendations that we could advise our patients of to help reduce the adverse events and improve outcomes in our clinic. So diet is the first thing to address because the gut microbiota is very malleable and rapidly responds to a change in diet. So a diet that includes a large variety of different coloured vegetables that are high in polyphenols and prebiotic will increase the number of beneficial species in the gut. And these foods really need to make up the majority of the diet the majority of the time. And most definitely we should be advising patients to avoid foods that are high in histamine and condensed proteins, no matter how pretty the packaging is. We really can't ignore the science. And then, of course, these are all common sense. We avoid allergens, processed packaged foods, additives, fad diets, and even the foods that look like they're health foods, but they're not. They're all just junk in disguise, and they can all contribute to dysbiosis and inflammation. So that sums up the diet, and I'll move on to supplements. So when it comes to supplements, probiotics seem to be the most common recommendation, and there's a lot of research to back that up. You can see here that in human studies with specific probiotic strains, there are some beneficial effects in the skin, including immune tolerance or improving immune homeostasis. And the same goes in the gut. There's a lot of beneficial effects there as well. But these effects are temporary because probiotics don't colonize the bowel, and prebiotics are actually better for that purpose in the long term. And studies usually use a specific strain of one single bacteria, whereas most probiotics available are multi-strain and they're unregulated, so you can't be certain of their action. And there's also the concern that probiotics contain molecular patterns that can initiate an immune response, and we don't want to do that if an individual has a disordered immune activity. So I think it's best to leave them out in our setting unless you have a really good understanding of how and when to implement them. But these are some supplements that you can recommend. Lots of benefits here. Just make sure that they don't have any additives, because most will have additives that can potentially contribute to dysbiosis. And also check that the doses are therapeutic, because you really do want them to be effective. And lifestyle changes are essential because overactivity of the HPA axis can override the benefits of diet and supplements. And that finishes up the presentation. The main takeaway points are that a certain percentage of our patients are likely to have some form of gut-mediated inflammatory process that contributes to skin aging and also increases their risk of an adverse event. So by providing these patients with some guidance on how to manage dysbiosis and inflammation, we may be able to improve outcomes for them in our setting. So I hope this has been useful for you, and if you'd like more information on how the microbiome influences our health, I've presented a series of webinars on the topic, and I'm happy to pass these on, so please get in touch. Thank you, and I will see you at the panel session.